now that you have an idea of what DNA is, we're going to talk a little bit about how advancing technologies today are using what we know about DNA to do a lot of different things, from solve crimes to manufacture hormones that some people need in order to survive. For example, diabetics who need insulin. We're going to learn how we can use DNA and our understanding of DNA to perhaps help fix genes in people. And this is all part of the process of biotechnology and the use of recombinant DNA. And we're going to go through each of these separately and talk about biotechnology first to start with. In essence, what biotechnology is any time I use a living organism or some sort of a cell part in order to make products. And, for example, this could be using bacteria to manufacture insulin for diabetic patients, which is something we do all the time. It might be altering tomato plants so they make better fruit that's more nutritious for us or that tastes better. And this is frequently done through genetic engineering, which simply means to purposefully alter the genes in DNA. And it sometimes also is done through recombinant DNA, which simply means taking a part of DNA from one organism and combining it with DNA from another organism. And I'll give you an example of that. Recently, researchers took a gene from a Atlantic flounder. A flounder is a type of fish found in the North Atlantic. It's used to very cold water, in fact, near freezing temperature. And they took the gene from the flounder that helped it remain alive and avoid cell damage at freezing temperatures, and they took that gene and inserted it into a tomato plant. And the upshot of that is that now they have tomato plants that are more resistant to frostbite when temperatures drop and are less likely to have tissue damage when it gets cold out. So biotechnology has opened up a whole universe of different things that we can do to DNA that we have in our environment and how we choose to use it. Now, recombinant DNA is nothing new, okay? This idea of taking DNA from one organism and combining it with another. It happens naturally all the time, and in fact, in my last lecture to you, I discussed several ways in which recombinant DNA happens naturally. I gave you the example of transformation and of transduction. And those processes, for example, are ways in which viruses can transport DNA from one bacterial cell to another, or how a bacterial cell can pick up DNA from a dead cell and utilize it. And then, of course, we talked about conjugation, where bacteria can share DNA with each other. And I also discussed transposons, those jumping genes that can jump from one section of DNA to another. Those two cause the production of recombinant DNA. We as researchers, though, have also been able to develop techniques to artificially combine DNA from two different organisms. And frequently the way this is done is to use those plasmids that are found in bacteria. Remember, plasmids are those little rings of genetic material that are external to the nucleus. So if this is my bacterial cell, and here's the bacterial chromosome here, the DNA, this plasmid represents another little section of DNA that's separate from the bacterial DNA. Sometimes researchers will use a plasmid as a way to insert DNA into another cell, or sometimes they'll even use viruses that attack bacteria specifically, and those are called bacteriophages. So we can use recombinant DNA for lots of things. I gave you the example already of using bacteria and inserting the human gene for making insulin into that bacteria and fooling that bacteria into making insulin for us that we can then purify and put in a vial so a diabetic can use that. Another example is we've also induced yeast cells, a type of fungus, to suddenly manufacture components of the hepatitis B vaccine, okay, again, through recombinant DNA. So let's look at an example of how we might go about making recombinant DNA. And the way we're going to do this is to start out with a plasmid. So here, again, in my bacterial cell, it has this hot pink plasmid in it. We're going to take that plasmid out. We're going to chop out a section of DNA from another cell. They're using a bacterial cell as an example here. And we are going to insert that foreign DNA into this plasmid. And the way that we do this is to use something called restriction enzymes. Restriction enzymes are enzymes that occur naturally in bacterial cells. They are there to help chop up DNA that invades them. For example, if a virus invades a bacterial cell, it brings its DNA with it. Those restriction enzymes will chop up that DNA so that hopefully they can destroy the DNA before the virus has a chance to parasitize the bacterial cell. Now, these restriction enzymes work in a very interesting manner, and yes, good, I do have it here. And I want to show you how that works, and then I'll jump back to the previous slide. Restriction enzymes will there are different types of restriction enzymes. Each different type of restriction enzyme is sensitive to a particular nucleotide sequence in DNA. So let's take this restriction enzyme, for example. It happens to be sensitive to the sequence CTAG. It will, whenever it encounters DNA, it will read that DNA, and any place where it sees the sequence CTGA, it's going to make a cut. Okay? It's going to cut just like this. And what I'm left with is two separate pieces of DNA, and notice that each of them have a region of DNA dangling without a complementary strand across from it. These are called sticky ends because they're very likely to stick to anything else that has the right complementary DNA sequence because that's a more stable form. So imagine if we have this plasmid and we use restriction enzyme to cut the plasmid at that particular sequence, and it does so. We're left with a sticky end here and a sticky end here. Then imagine we get some DNA from another organism. Let's say it's a human gene for making insulin. And we use the exact same restriction enzyme on it. It's going to make the same kind of sticky end cut any place it sees that same CTAG sequence on it. What's nice about that is now we've opened up this plasmid. It has these sticky ends, and the sticky ends are complementary to the sticky ends we just created on the DNA in yellow here. They will anneal or stick together quite nicely, and what we've just effectively done is inserted another organism's DNA into this plasma. In other words, we've just made a piece of recombinant DNA. Now I'm going to back up here. That's this step here. Here we've inserted that recombinant DNA into that plasmid, and then we can then take that plasmid and perhaps, I don't know, we could, in this example, for example, insert it into a plant, and as a result, the plant would carry whatever genes we had inserted onto that plasma. Okay? So this is a very effective technique for introducing genes into other organisms. There is a great animation on restriction enzymes and cloning online in the classroom in this week's learning unit. You need to go through it until you understand it, so don't skip that. It's very important. Now, plasmids are very frequently called vectors because they are a way to transport, in this case, genetic information from one cell to another. We can either use a plasmid to be a vector that transports DNA from one organism to another, or we can use a virus to do that transport. More frequently, at least for our discussion here, we're going to focus more on the plasmids. This is an example of a plasmid, a real plasmid from a bacterial cell, and it shows you the sequence of all of the different types of restrictive enzymes that would cut it. Remember, I told you that there are lots of different types of restrictive enzymes, and they all look for a different sequence of nucleotides. In this plasmid here, the E. coli plasmid would cut it there. By comparison, the, let's see here, E. coli would also cut there, okay? A different restriction enzyme would cut here and here. Okay, so it just depends on the different restriction enzyme where you would cut in the plasmid. Sometimes that can be very useful. Just remember, again, restriction enzymes are naturally occurring enzymes found in bacterial cells. They're there to help combat viral infection, but we as humans are now using those restriction enzymes in a laboratory setting to help us insert foreign DNA into a plasmid. Okay? Then once we have this plasmid inserted into a bacterial cell, if we let that bacterial cell divide and divide and divide, what we are in essence doing is cloning or making millions of copies of that plasmid. Okay? If the plasmid was in this cell, then it will be in both daughter cells, and then it will be in all 4 of these daughter cells. So this is an effective way of cloning or making large number of copies of a particular sequence of DNA. This is kind of the old-fashioned way of copying DNA. We now have a much more effective and useful way of copying DNA called PCR, or the polymerase chain reaction, and that's what I'm going to talk about in the next lecture in this series.